Good morning, everybody. My name is Zhang. I am the scientific staff here in LMB, specialized in flow cytometry. Today, I'm going to give you an introduction to flow cytometry. You can put your questions in Q&A box, and I will answer as much as I can at the end of this talk. So in this talk, I'm going to introduce our flow cytometry facility and talk about what is flow cytometry, how does a flow cytometer work, sample preparation, and some examples for applications that has been done here in LMB. There are three of us. Maria is the manager, me and Ricky. We're looking after two labs. The main lab accommodate all our sorters and some analyzers, and the smaller lab accommodate two high-end analyzers. For full specification for our instruments and the colors that you can do on those instruments, please refer to our internal website. In brief, we have 6 analyzers. 4 of them are high-end analyzer as showed in here. They all can look up to 10 and more colors at the same time. We have a simpler cytometer called Sony Eclipse. This is calibrated to look at cell volume, also do the cell count. We have a Sony spectral analyzer. This uses a novel spectral technology to look at the full spectrum of each fluorophore we use. We have 4 cell sorters, 1 MoFlow high-speed cell sorter, 3 Synergy high-speed sorter. We will soon have a new addition to our cell sorter, which is a FACS aerial fusion. To book a sorting time, please contact our staff by email. To book analysis time, please use Cario Connect. So I'm going to start with the definition of flow cytometry. Flow cytometry is a laser-based technology that used to measure and detect the physical and chemical characteristics of cells or particles. In this process, a sample containing cells and particles is suspended in fluid and injected into a flow cytometer instrument. Ideally, the cells passing through a sensing point one by one. Flow cytometry measures fluorescence intensity produced by fluorescent conjugated antibody or the fluorescent ligands that binds into internal molecules such as DNA. Flow cytometry can analyze the expression of extracellular and intracellular molecules. It characterizes and defines different cell types. Analyze cell size. It allows simultaneously multi-parameter analysis of single cells. So what is the advantage of flow cytometry technique? When we compare image cytometry and flow cytometry in terms of localization of antigen and numeration of cell types, and we can see the value of this technique is the capability of measuring a large number of single cells in a very short period of time to give us quantifiable data. So how does a flow cytometer work? I'm going to start with fluorophores and fluorescence, also talk about components of a flow cytometer. Fluorophores are molecules that can be excited by external light. It rises to a high energy state, however unstable. It loses energy and emits light at longer wavelength when returning to ground state. The process of excitation and emission produces fluorescence. It's worth to mention that the emission wavelength always longer than excitation wavelength. This produces a Stokes shift. So what are these fluorophores that we use in flow cytometry? The first one is fluorescent protein that discovered in nature. So, for example, the green fluorescent protein was discovered in jellyfish. This natural protein can be genetically engineered as a fusion to other protein, act as a marker. We have many fluorescent protein with different color to use in flow cytometry. Use of this fluorescent protein allow us to quantify intracellular markers in live cells. Second one is single dyes. These are the long-used single dyes. For example, FixAP, APC, and PersAP. These single dyes act as base for tandem dyes. Tandem dye comprises a small fluorophore that's covalently coupled to another fluorophore. When the donor fluorophore gets excited, the emission wavelength overlaps with excitation wavelength of acceptor fluorophore. The acceptor fluorophore emits light in a longer wavelength. These are the examples of tandem dyes we commonly use in flow cytometry. For example, P-Cy5 that is derived from PE, APC-Cy7 that is derived from APC. So why we use tandem dyes? In a limited amount of biological samples, we always want to increase number of parameters that can be detected at the same time. Tandem dyes make it possible to expand our panel design. For example, the range of BD brilliant violet dyes emits light at different wavelengths, as shown in here as different color. They all can be excited by the single violet laser line. Similarly, the BD ultraviolet UV dyes can also be excited by a single UV laser line. So what are components of a flow cytometer? A flow cytometer comprises laser lines, fluidic system, optics, detectors, and electronic system. A fluidic system is to transport cells or particles in a fluid stream to the interrogation point of a laser. Ideally, these cells travel in the center of the laser beam. Only one cell or particle passes in the interrogation point at one time. The fluidic system requires stable running of fluid stream, free of bubble, dirt, and clumps. Depends on the laser arrangement. The cells or particles passes only one interrogation point on a collinear laser line. They pass several interrogation points when it is a spatial separated lasers. So to ensure the cell travel in an organized way, heterodynamic focusing is the method that utilizes a fast-moving sheath fluid, force the sample into a very small core stream, causing cells travel at the same speed, arriving at interrogation point in a single cell line, in a single file line. The sample stream does not mix with sheath stream in principle of laminar flow. So sometimes in order to increase sample input rate, the sample core is widened by increased sample pressure. So the widened sample core comes with consequences. the data shows broadened distribution, decreased resolution. The signal can be aborted because of the increased coincidence. So for optics, the solid laser lines are often used as excitation source. These are 5 laser lines we commonly use in our flow cytometers. We often use the color to name over the wavelengths of lasers. The lenses are designed to shape and focus a laser line onto the sample stream. Optical mirrors and filters are used to direct and separate emission light. The first two are the two types we can see in our flow cytometer most of the time. Dichroic mirror act as beam splitter. They reflect certain wavelengths at certain direction and allow remaining wavelengths to pass through. The bandpass filter allows only a specific limited bandwidth to pass through while rejecting all other wavelengths. So this is an example of optical pathway. The cell marked with different fluorescence markers passing through the laser beam. The fluorescent marker emits light. So take a fluorescent marker PE as an example. PE excitation wavelength max around 585 nanometer. So the light around 585 nanometer pass through the first dichroic mirror, and second dichroic mirror, because it's longer, gets deflected by third dichroic mirror 620 because it's shorter. The PE signal gets deflected to enter a bandpass filter. The bandpass filter here is 585/40. This allows the bandwidth between 585 minus 20 and plus 20 to pass through. So the PE signal can be filtered, reach the detector. In this way, the PE can be separated from other fluorescent markers. Spectral technology is considered as next generation of flow cytometers. Different from conventional cytometer, the spectral analyzers use prisms to split the excitation light across array of detectors. In our spectral analyzer, we have 32 channels to capture the whole spectrum for each fluorophore. So this technology is able to separate highly overlapped fluorophores, also enhances the detection for dim population. The electronics are used to convert light signal into data. So the electronic detectors convert incoming photons to voltage pulses. So these pulses are then converted to a digital number by analog digital converter. The digitalized number and then transferred to computer, stored as data for us to analyze. So how the voltage pulse is generated and measured. As the cell enters the laser beam, the voltage pulse starts to rise above the baseline. The pulse peaks as the cell enters the laser beam completely. As the cell exits the laser beam, the signal starts to decrease and eventually drop to the baseline. The width of the pulse corresponds to the time of the cell in the laser beam. The height corresponds to the light intensity from the cell. The area is the integral data combined from width and height. So how to interpret the flow cytometry data? I will start with the scatter signals. So as the cell passes through the laser beam, the cell scatter the light in all directions. Forward scatter collect the scattered light at forward direction. The magnitude of a pulse is roughly proportional to the size of the cell, which means the small cell generates a small pulse, the large cell generates a large pulse. We can plot the forward scatter signal in a single dimension to produce a histogram. As we can see the distribution of related cell size. So the set scatter collects the scattered light from the 90-degree angle. The set scatter is caused by granularity and complexity inside of cells, also on the surface. So increased granularity of a cell results in a larger scatter light, hence a larger voltage pulse, as we can see here. However, sometimes the profile in histogram cannot define different cell types. If we combine the cell size and granularity, plot each single data point in a 2D plot, the cells with the similar size and granularity cluster together, as we can see here. So we need to know our cells before analyzing them. We need to know what kind of cells we are expecting, and are they homogeneous or heterogeneous, and their size. So make sure they fit the flow cell or nozzle you use. Threshold is set to ensure we collect useful information and ignore the noise. By cutting off the irrelevant signal, the population of interest can be clearly defined. The same principle applies to fluorescent signal. The fluorescence conjugated antibody detects the antigen. So the fluorescent intensity is proportional to the level of protein expression. The least expressed antigen produces a small voltage pulse. The largely expressed antigen produces a larger pulse. If we only use one fluorescent marker, we can use histogram to review the level of protein expression and calculate the percentage of positive population. However, in most cases, we use many more fluorescent markers in a sample. So the 2D flow cytometry plot is more useful. So as we can see, use a white blood cell surface marker against set scatter. We can define the different cell population from white blood cell in the white blood cell population. So we can gauge the population of interest, such as lymphocytes, to study the subsets. The data acquired from flow cytometers are stored as a standard FCS file. So these FCS files can be analyzed offline by independent analysis software such as FlowJo and FCS Express. These are the commonly used statistical tools in data analysis. For example, percentage total or percentage parent can use to calculate the percentage of positive population. The median or mean fluorescent intensity can be used to compare the level of expression across different conditions. The power of flow cytometry comes with cell sorting. According to the different fluorescent markers on the cell, we can define the population of interest. These cells are isolated by electronic static sorting. So this purified population of cells can be subcultured or applied to further analysis. This is the view of our no-flow cell sorter. In the upper chamber, we can see lasers intercept sample stream. This stream is vibrated at very high frequency. A 70 micron nozzle can generate 78,000 droplets per second. We can sort up to 25,000 events per second. In the lower chamber, there are a pair of deflection plates. The stream droplets pass down through those charged plates and deflected into collection tubes or plates. We can sort into different sizes of tubes, plates, anything that fit into our collection device. So a sample preparation is a key to getting good data. A single cell suspension is necessary in flow cytometry analysis. So some cells come naturally in suspension, which will be easier to handle. Some other cell lines grow adherent on plastics. So we need to dissociate these cells by appropriate reagents. Tissues are more difficult and need mechanical or enzymatic dissociation. Some cell strainers with different size help to remove cell clumps. So the sample we get should contain as little debris and as few clumps as possible. There are other things to be considered. For example, dilute your sample to a concentration that instrument can handle. Get rid of unwanted cells. For example, lyse red blood cells, fecal cells from blood and tissue to separate mononuclear cells from granulocytes. It is always wise to include a viability dye to get out dead cells which produce non-specific binding. Always includes controls. The instrument is set by users, so you need to choose the parameters appropriate to detect your fluoroform in the sample. Use negative control to set your background values. Use single color control to assess the spillover and perform color compensation. These are the basic rules for panel design. Brightest dye on least expressed antigen, choose dyes with least spectral overlap, or use dyes that are excited by different laser lines. So what is spectral overlap? Take example of Alexa Fluor 488 and the PE. We can see the excitation wavelength from the Alexa Fluor 488 is going into the PE detector. So this caused a spillover from Alexa Fluor 488 into PE, resulting a false signal in PE channel. The single color control is necessary for each fluoroform that you use in your sample. For example, we run Alexa Fluor 488 control. The spillover from Alexa Fluor to PE can be assessed and compensated, can correct by compensation. So in reverse, the PE control can be used to assess the spillover into Alexa Fluor 488 and correct by compensation as well. So these are some examples of flow cytometry application that have been done by LMB scientists. So we can see the blue fluorescent protein and the green fluorescent protein that are fused to a protein plasmid report the protein expression level in cell lines. So some cells is more homogeneously expressed than the other. The fluorescent protein can be also used in mouse models. So this is example to show 4 different color of fluorescent protein reporting 4 distinct transcription factor that has been engineered into a mouse model. We also can study apoptosis using flow cytometry by using a live and dead marker, which is PI, and also annexin 5. Annexin 5 binds to phosphatidylserine that is translocated on external cell membrane on apoptosis cells. So we can see the dead cells are double positive for PI and annexin 5. The apoptotic cells are only positive for annexin 5. So we also can study cell proliferation using tracing dyes, such as CFSE. The cells are loaded with CFSE. As cell divides, the fluorescence is halved. So after a length of time, we analyze the sample. The CFSE fluorescent intensity reveals the cell division profile. So this is an example to show you the T cell proliferation from a mouse spleen. So we use cell trace violet to load the cells. On the day 4, these T cells can be seen gone through a several cell division. So we also studied L13 in cell division. We can see the expression of L13 start to increase as cell divide, indicating the development into H2 cells. So there are a number of DNA binding dye can be used to study cell cycle. This is example to show the cell cycle in retinal pigment epithelial cell line. The cells are stained with Hurst. So the Hurst fluorescent intensity reveals a cell cycle profile. As we can see clearly, the G1 phase, S phase, and the G2 phase. In this study, a cell surface protein also be investigated in the cell cycle. I have to mention that it is very important to remove doublets and clumps in cell cycle study because, as you can see, 2 G1 cells stuck together pass through the laser beam. The signal generated can be falsely regarded as a G2 cell. So if we plot peak and against area signal, the single cells can be gated to reveal the genuine cell cycle profile. This is another example to show you the cell cycle in haploid cell line. Haploid only contains one set of chromosome, so you can see these haploid cell line become diploidized. So the diploid cells undergo a cell cycle. BRDU is used to assess DNA synthesis in cell cycle. BRDU is a thymidine analog incorporated into DNA as cells enter S phase. So this S phase can be identified by detecting BRDU using fluorescence-labeled antibody. So this example comes from the paper that has been published by one of our users. The wild-type MEF cells shows a typical cell cycle profile with distinguished S phase. The depletion of a protein in this cell line caused the genome instability, so these MEF cells become polyploid. An abnormal amount of DNA can be observed in these cell lines. Flow cytometry is capable of karyotyping of cells. So this chromosome was isolated and stained with dual DNA binding dyes. The chromosome can separate according to DNA size and composition. So we can see these are 23 copies of DNA and containing a Y chromosome. This is a human male cell line. Immunophenotyping is a big part in flow cytometry studies, which involves antibody staining. It is necessary to titrate your antibody to find the optimal condition for use. Start with the concentration recommended by the manufacturer. A series of concentration of antibody are prepared by doubling dilution. So incubate your cells with this range of concentration, run a sample on a flow cytometer, gauge the positive and negative population, calculate the median fluorescence intensity. If we plot the ratio of median fluorescent intensity of positive population over the negative population against the dilution, the optimal concentration can be found at the saturation point. So this is an example to show you the human whole blood phenotyping. So we can see by a white cell surface marker and cell scatter, we can find different cell types. These cell types can be further investigated. We can see the different type of T cells in the upper panel. The lower panel shows you the dendritic cells, natural killer cells, and the B cells. The different type of progenitor cells differentiated from hemopoietic stem cells also can be classified using its distinct surface markers. So our user Huang has published this Nature paper, which involves a large amount of flow cytometry work. We sorted a single hemopoietic stem cells from a genetically deficient mice according to its surface markers. These single HSC cells are then transplanted into a blank mice. 16 weeks later, we sort again to isolate these HSC progeny. These cells are further studied for its functional capacity. The intracellular cytokines can also be investigated using flow cytometry. The sample prep often involves fixation of cells, permeabilization of cell membrane to allow antibody gain access for intracellular targets. So this example to show you the cytokine expression in T cells. So the T cell, so the first upper panel is the control. So on the day 7, the CD4 T cells undergo a several cell division, as you can see in the green plot. The 2D dot plot tells us interleukin-2, L2, produced from first T cell division onwards, and interferon gamma produced after a number of cell divisions. We can also study calcium influx using flow cytometry. So INDO1 is a ratiometric calcium dyes. This dye can be excited by UV laser. When bound to calcium, it emits at 405 nanometer. When unbound, it emits at 475 nanometer. So this is example to look at calcium influx in naive T cells. The naive T cells firstly defined by its surface marker. And then an antibody linker was used to stimulate calcium influx in naive T cells. So the calcium concentration increases in the cytoplasm. INDO1 emits more violet light than green lights. When we plot the ratio of violet light over green light against time, we can clearly see the activation of naive T cells in real time. So the above slides are all the information I'm going to share with you today. These are some reference books and websites for anyone who wants to do flow cytometry assays. Please refer to our internal website for more details. We're always happy to talk to our users regarding training on instruments, cell sorting, flow experiment design, fluorophores, any possible applications using flow cytometry. We have flow cytometry clubs and societies regionally in the UK-wide and worldwide. You're welcome to join us for flow cytometry meetings. So finally, thank you very much for your attention. So please put your questions into Q&A box. I will answer your question as much as I can. Thank you. Okay, so I have one question. So how does the software calculate compensation? So compensation is a algorithm. So it's a mathematical calculation. When you run your single color control, so the software can assess the percentage of the spillover from one channel to the other. So they subtract certain percentage of the signal that's built into the other one and correct the signal just by subtraction. So this is done automatically by the software. So there's another question. If my overexpressed protein has only HA tag in N-terminal, is there any possibility to sort the cells on the expression level? Yes, if it's a fluorescent tag, so or you tag if you target this tag by the antibody, as long as there's a fluorescence, so we can detect the fluorescence if they appears expressed at different levels, so we can gauge on the different intensity of the fluorescent. We can gauge the different fluorescent intensity to sort these cells. Yes, it is possible. Okay, do we have other questions? Okay.
Okay, do we have any more questions with the, otherwise, thank you very much for the talk and thank you for your questions, and look forward to seeing you next Tuesday, which is bioinformatics.